Dr. Akbari received his bachelor's and master's from the University of Nevada at Reno before completing his PhD at the same institution in 2008. He then joined Bruce Hayes' lab at Caltech as a postdoctoral scholar before he was appointed as the assistant professor in entomology at the University of California, Riverside. And at present, he is a food professor at the University of California, San Diego, where he works on researching different genetic strategies to limit the spread and control of mosquito-borne diseases. As somebody who's grown up in Singapore myself, I know many people who have been impacted by mosquito-borne diseases, including myself, and I'm incredibly excited to see how Dr. Akbari is using synthetic biology to tackle this very pressing challenge in our world. And without further ado, Dr. Akbari, if you would like to begin your talk.
Great. Thank you so much, Lee, for the kind introduction. And also thanks to the organizing committee and Alex for the invitation to be part of this Gogec, I'm not sure if I'm pronouncing it right, Gogec conference. I know this is a new thing, so I'm really happy to be part of it. And I hope you enjoy my talk today, which is going to be describing how we can use principles of synthetic biology to eliminate human disease vectors. All right, so let me okay, so I'm just going to give you a little overview of what I'm going to talk about before I begin. So I want to talk to you first about sort of what types of pathogens mosquitoes actually transmit and how we currently control them. And then I want to sort of lead into sort of some of these advanced genetic engineering techniques we can use to control mosquitoes, which rely on principles of synthetic biology. And in particular, I want to talk about two things. The first is going to be gene drive, which I'm sure maybe some of you have heard about this because it's been kind of highly discussed in the last several years. And then I'm going to lead into sort of alternative techniques of genetic control using a system that we termed precision guided sterile insect technique. And I'll tell you what that means when we get there. So I always love to start with this slide. This is a slide from Bill Gates. And it just sort of emphasizes the impact mosquitoes have on humanity. If you think about the world's deadliest animals, you hardly would think about the mosquito. But in reality, the mosquito is actually the world's deadliest organism. It kills more humans on Earth than any other animal combined. And the next closest thing is humans killing each other. So why are mosquitoes so deadly? And I'm sure some of you, most of you probably know they transmit diseases. Malaria is the biggest one. We see about 207 million cases per year and around 1,000 deaths per day, even today. And most of these are children under the age of 5 in sub-Saharan Africa. And this equates to about a children dying every 2 minutes, which is pretty horrible. Mosquitoes also transmit a number of viruses like DENV virus, West Nile virus, ZIKA virus, yellow fever. And it's estimated that literally half of the world is at risk of being infected by a mosquito-borne pathogen. On the bottom here are just some of the major mosquito vectors that transmit these pathogens. I did this really interesting interview with a journalist from the LA Times, David Lazarus, earlier this year. If you have time, you can check it out. But it sort of links COVID with mosquitoes. And we talked a lot about how when COVID first came on the radar, a few labs did tests to make sure that mosquitoes can't transmit COVID. And it turned out that they could not. But we talked about like what would happen if they could, right? And we got really lucky. We dodged a bullet here with mosquitoes not being able to transmit COVID as well. I think it would have been a much worse pandemic. And if you think about our last sort of epidemic, which was Zika virus, that was transmissible by mosquitoes. So we really need to develop new technologies to prepare for potentially the next pandemic, which may be mosquito-borne. We don't know. All right, so how do we protect ourselves from mosquitoes? And really our first line of defense is to reduce contact, right? And we could do that by using repellents. We can use bed nets at night when we sleep. There are also these lures and traps, which aren't that great, but they also work somewhat. We can modify the environment. Mosquitoes lay their eggs in standing water. And so if you could reduce the prevalence of standing water, then you could reduce mosquito populations in that way, simply by, like in your yard, removing breeding sites like, flower pots or tires and things like that. Insecticides and larvicides, these things are effective, but they're also costly, and they have other issues of mosquitoes evolving resistance to them. Pathogen removal drugs, there are things like artemisinin, which exists for malaria, that are on the market. And then we have these newer technologies, which are the genetic technologies. And in the United States now, you may have heard that Oxitec, which is a biotech company based in London, is actually releasing mosquitoes in Florida at the moment. And they're doing this as an experimental trial to see if they can reduce populations in Florida. Now, while all of these different practices for mosquito control are essential for preventing disease transmission, none of them are perfect, right? They all have issues. Repellents, bed nets, these require continuous application. You have to keep applying them. And you also have to distribute them to people that need them. Modifying the environment, it works, but it's difficult, right? It's very hard to reduce the breeding site, especially for mosquitoes like Aegypti aegypti, which breed in very small containers, like even a Pepsi bottle cap, they can breed in that. So how do you stop that? Insecticides, larvicides, these are expensive, and there's been evidence of evolved resistance to them. And then finally, the genetic technologies which are used today are just not sustainable. So we need new technologies, really. I wanted to point out that the mosquito is an obligate vector for pathogen transmission. What I mean by this is you have a wild mosquito, it's going to go bite an infected individual, take up the pathogen, incubate it, and then transmit that to the next unlucky individual. So how do we block this very simple chain of transmission? And really the holy grail is to develop effective vaccines. If we can vaccinate everybody for dengue, malaria, Zika, etc., then you wouldn't have this problem. An alternative method would be to engineer mosquitoes that are pathogen resistant. And this is what I'm going to talk to you about today. So over the last 20 years or so, there's been some really exciting research in terms of engineering mosquitoes that are unable to transmit pathogens. And here are just a few examples of work done on the top, led by Tony James, where they've actually shown that they can prevent mosquitoes from transmitting the malaria parasite. And then on the bottom, there's an example of a system that is using RNAi to prevent mosquitoes from being able to transmit dengue virus. So when I started my lab, sorry, I skipped a slide. So when I first started my lab at UC Riverside, it was kind of an exciting and scary time. It was exciting in that, CRISPR was really accelerating. People were really starting to use it big time. And so our lab really adopted that. Additionally, it was right at the time when ZIKA virus hit. So there was a lot of interest in understanding what ZIKA virus was and how it's being transmitted and how we can stop it. And so, one of the first projects I took on was to use principles of synthetic biology to see if we can engineer mosquitoes that can be able to block their ability to transmit ZIKA virus. And so in collaboration with Prasad Padakar and Chenhong Chen, we developed this system here. So on the right here, you can see these transgenic mosquitoes. These express this fluorescent protein in their eyes, so you can easily distinguish them. And so what did we do? So basically what we did is we looked at the ZIKA virus genome, which was sequenced at the time. So we had the sequence of that, and it was about a 10.8 kb single-stranded positive sense RNA, and encoded a single open reading frame that gets processed into about 10 different proteins. And so with that sequence, what we did is we went in and we designed these microRNAs and synthetically engineered them to target different regions of the Zika viral genome. And so on this image here, you can see sort of which genes we're targeting. And so we had a polycistronic array of 8 synthetically engineered microRNAs that are all expressed from a promoter that gets activated when the mosquito takes a blood meal. So when the mosquito gets a blood meal, this promoter comes on, it expresses these microRNAs in the midgut, which is where the blood comes in. So if the virus is present, then these can go in and sort of block the replication of that virus. So we designed this and we engineered the mosquitoes, and then we basically challenged them. We challenged them with ZIKA virus. So this was work done at CSIRO in Australia by Prasad Padakar. And basically what he did is he used wild type control mosquitoes, infected them with Zika, and measured their, the viral titer inside of them, which was quite high. And then he did the same experiment with our anti-Zika mosquitoes and found that they were unable to transmit the virus. So we're looking at transmission, and the way we do that is by actually assessing viral titer in the saliva. So in the saliva, there was no Zika virus present if we encoded this cassette in the mosquito. So this was a really interesting experiment. And then we kind of went on to this experiment here. And so dengue virus is actually transmitted by Aedes aegypti mosquitoes, and there's 4 different serotypes of dengue virus. But there was no real effector that can block all serotypes. So there was some work by James Crow, which found a broadly neutralizing monoclonal antibody in humans that can protect against all 4 serotypes of dengue virus. So what we decided to do was to encode that that monoclonal antibody and re-engineer it as a single chain antibody and express it in the midgut in the mosquito, with hope that we can prevent transmission of these 4 different serotypes of dengue virus. So we did this. So on the top, you can see we have this promoter that's, again, active in the midgut in response to blood meal. It expresses single chain antibody. We have a marker. So these have actually red eyes, so we can detect them. And then we basically did this challenge assay. And what we found was similar to the Zika virus experiments, in that in the saliva, if we encode this single chain antibody against the different serotypes of dengue, we can sort of block transmission of each of those serotypes. So this is really exciting work. But really what I wanted to show you with these experiments is that these observations tell us that we can actually completely harness the mosquito immune system and prevent them their ability to be able to transmit malaria, dengue virus, and Zika virus. All right, so if we can do that, right, we can harness their immune system, we can engineer them using principles of synthetic biology. Why don't we now take these mosquitoes that cannot transmit malaria, for example, or Zika or dengue, and why don't we just release them into the wild? And with the goal of replacing the wild mosquitoes with these engineered mosquitoes that cannot transmit disease. And sort of on the bottom, what we would hope is that we would have less transmission by mosquitoes, which would equate to less human disease. So this is how I think about this conceptually. So you have a factory, you engineer your mosquitoes that are engineered to block transmission, and you sort of master them, and then you release them into the wild. And then you have your disease transmitting mosquitoes in the wild. And what you want to happen is you want these genes that are encoded in the mosquito to transmit into these wild populations rapidly, right? Because you don't want evolution to take over. So you want it to happen fast. And so, hopefully within 1 to 2 years, all mosquitoes in the wild will now be unable to transmit diseases. And, we will solve sort of a major world issue, right? That's kind of the goal. All right, so the grand challenge here is, how do you force a pathogen-resistant gene that's encoded in a mosquito that probably encodes a fitness cost to spread itself into a wild disease-transmitting population and basically engineer the population to be pathogen-resistant? How do you do that? And the underlying problem here is a problem in genetics. And the problem is that these genes that encode pathogen resistance, either the anti-Zika, the anti-dengue, the anti-malaria, which I showed you, these result in a fitness cost for the carriers. So those animals are not as healthy as like wild mosquitoes, right? Because they've been engineered, they've been reared in the lab. They encode these fluorescent proteins, these single chain antibodies, which probably impact the fitness of the animal. So how are they going to compete with these wild mosquitoes? Well, the answer is that they're not going to be able to. But there's a solution to this problem. And the solution is to link those genes with a gene drive system. And the goal here is that, when you do this, the gene drive systems have this ability to be able to spread into a population even if they decrease the fitness of the animal that they reside in. So over the last, let's see, I've been working on this since like 2009. So over 10 years, we've engineered a number of different population control technologies, including gene drives. And pictured here are just a list of the ones we've generated. We have Medea, we have underdominance maternal effect lethal, we've engineered reciprocal chromosomal translocations, we've engineered reproductively isolated species, and we've also worked on CRISPR homing gene drives. And all of these are different technologies that can be adapted to different species to control populations. But today I'm going to talk to you about only two things. And on the bottom, we're going to talk about a confinable CRISPR homing-based gene drive. And I also want to talk to you about a new system that we developed, which we call precision-guided SIT. All right, so CRISPR-Cas9 gene drive. I'm sure many of you may have heard of this. It's been widely discussed, and it's something that has potential to, prevent deadly diseases like transmission of malaria, for example. And that's kind of one of the leading potential uses of gene drive that's being discussed. But the truth is that, CRISPR, it did accelerate the development of technologies like gene drive, but gene drive was a technology that existed far before CRISPR, right? And I was building gene drives before CRISPR, but others were also using things like TALENs or zinc finger nucleases to target precise sequences in genomes. But of course, CRISPR is just so easy to use, right? Especially with Cas9, it works so well. And if you encode to build a gene drive, it's very simple in that all you have to do is basically encode Cas9 to be expressed in the germline. You have a guide RNA that's going to also be encoded in the germline, and it's going to be positioned opposite its target. And then you could put a cargo in there, and that cargo could be an anti-malarial effector gene or anti-dengue, anti-ZIKA, whatever you want. And that cargo will sort of transmit along with the gene drive. So basically you encode these components in the genome and have them expressed in the germline. And then when you have a heterozygote, what happens is that animal is going to mate to a wild type. And in the germline, you get this copying happening where the Cas9 is going to go cut the opposite allele. And basically you get homology-directed repair, which enables copying of what's in the gene drive over to the opposite allele. And that basically creates it turns a heterozygote into a homozygote. And what that does over time is that as that keeps going into the population, you keep converting heterozygotes to homozygotes. And when they mate out, they make more gene drive animals. And then eventually, you get quick spread into a population. Now, this technique can be useful for two types of systems. One system is called population modification. And what this is basically you release your gene drive into a population and it spreads and continues to spread until it reaches fixation. And when it reaches fixation, the wild population still exists, but it's been engineered to be disease resistant, for example, if that was the purpose of it. So ecologically, the population still exists, but it's been engineered. And in principle, if the gene drive works perfectly, which so far none do, but if it does, a release of a single organism could actually make this happen. The alternate technique that's being used for population control with gene drive is population suppression. And the way this works is that you release a small number of individuals, they spread, and then they hit fixation. And when they hit fixation, the population declines and then eliminates or eradicates itself. And in principle, this could also happen from a single organism release. But it's different than replacement ecologically because population suppression, if it works, at the end, the population no longer exists. So those are the two broad categories of gene drives. And there's been a number of different issues that have been sort of brought up, especially with the issue of from a single organism release, you can get this effect. This results in the system being very invasive. If you release it in one location, it can spread all over, which can be a problem. It also makes it uncontrollable. it makes it potentially non-reversible. You would need to release a new gene drive to counter as a countermeasure. This makes it very difficult to measure risks, right? How do you test for risks if you can release a single organism and it's going to spread to fixation potentially? This also makes it very difficult in terms of regulation. Is this regulatable? What are the unintended consequences? Will the public accept this? Who decides if one could, release this and use this in their country if it's going to spread everywhere, right? And these are just a minority of issues that are being discussed sort of actively by both ethicists and regulators and stakeholders. So because of these issues, our lab has sort of been working more toward developing technologies which are controllable. And we do this by designing them to be self-limiting. And this creates these features of confinability, reversibility. It makes it more safe, and it still makes it effective. So I'll talk about what I mean by this. So one very simple way of turning a gene drive that is non-confinable, that can spread sort of uncontrollably is really, so if you look on the bottom here, we have a linked drive design on the left where Cas9 is encoded in the gene drive. But if you simply unlink Cas9 from the gene drive, you make what we call a split drive, then you generate this feature of it being self-limiting, where you have to do multiple releases for it to spread and fix itself into a population, but this gives you the control. Okay, so what our lab sought out to do was to develop a confinable gene drive in Aedes aegypti. Aedes aegypti is the major vector for dengue, chikungunya, yellow fever, and Zika virus. It's present in the United States. It's invaded California, so it's in my backyard. My kids get bit by Aedes aegypti. So it's presenting a risk here in the United States for transmission of these deadly diseases. So we wanted to develop our system in this species. And what we did is we basically encoded our drive and separate from the Cas9. So we have to do a genetic cross to actually measure for efficacy of our gene drive. But again, this gives us a split drive system that we want. So I'm going to spare you of all the details of how we actually did this. But basically, in this here, you can see this is what our system looks like. We encoded a marker gene and we targeted a I phenotypic gene called white. So we have a 3xp3-TD tomato, which is a fluorescent protein that marks our transgene. And then on the left here, you can see this white I phenotype. And as you go over, you can see a wild type phenotype of a mosquito has a black eye, and then of course no transgene present. So we can measure all these different phenotypes to see if our gene drive is working the way we expect it to, basically. And what we found is, in summary of what we found is this here. So we were able to generate a system that when we have a trans heterozygous female that encodes the Cas9 and the gene drive, and we cross it to a wild type male, then basically we get 84% on average of the progeny receive the gene drive. And then we also found that we get 100% cleavage, which means that this white eye phenotype, we get this this phenotype when we cross to Cas9. So, and that's because of this feature we call mosaicism. So when Cas9 and the guide RNA come together in the organism, you get expression throughout development, and then you get a mosaic animal that's arrived. So we get 100% of the animals with this mosaic eye phenotype, and then 84% of them actually get the transgene. So given those parameters, what we did is, in collaboration with John Marshall Lab at UC Berkeley, we did this model here. And what we were looking for is to see whether our system is effective using a model, and is it confinable? Does it spread into neighboring populations? So in this graph here, what we did here is we did 10 releases of our gene drive into a population at a 1-to-1 total population frequency for each release. And what happened is this red line, we follow that red line, you can see that it increases in frequency after the 10th release and is almost near fixation. And then it persists there for above 85% for 3 years, and then it starts to decline after that. And then in a neighboring population, it doesn't spread into that population. So if you compare this to, let's just release the refractory gene, so the anti-malarial effector gene, into a population by itself with no gene drive, and you do 10 releases of that, what happens is that goes up, it gets into decently high frequency, and then it falls out really quickly because it doesn't have a gene drive linked to it. And again, it also doesn't go into a neighboring population. Now, if we compare this to a linked gene drive, so if we had the Cas9 linked to the guide RNA and the gene drive design, and we did a single release, what would happen is that would go up in frequency and then persist for quite a while, but then it would also spread into a neighboring population. And that's the underlying issue, is that if you have this linked design, it's going to spread beyond your release site into other locations. And is that going to be a problem for acceptance and regulation going forward? We don't know, because nobody has done a gene drive release in the field, but time will tell. All right, so now our gene drive, we have a gene drive system. It works moderately well, right? It's 84% transmission, not 100%, but 84%. So we're ready to link an effector to it. That's kind of one of the things we're working on. Okay, so on this part of the talk, just a quick summary. So I've shown you that we've been able to engineer using principles of synthetic biology, Zika and dengue resistant mosquitoes. We were able to, of course, develop our gene drive system, which required us to develop Cas9 expressing lines and functional promoters in Aegypti, which was not trivial. And our split drive actually, it works moderately well. We have 84% transmission rate. And we were able to demonstrate drive stability over multiple generations. So we're working on some long-term population cage studies with the system. We want to link effectors to it. And of course, we're also working on generating new gene drive designs that are evolutionarily stable. And I'll talk about what I mean by that. So one of the issues with gene drive is, in addition to our system only transmitting to 84%, we also saw resistant alleles forming. And resistant alleles can basically completely block the spread of your drive in a population. And if we're seeing it in our lab, in a small little population in a few generations, what do you think is going to happen in the wild when you release this? When you have the diversity that's out there and you have evolution kicking in, it's probably not going to work. So what we've been working on is trying to improve the design to make it more stable. So we came up with a system which we call Homer, for home and rescue. And in the lab, we joke about, it's just we named it after Homer Simpson, but it's not really true. But I kind of threw that in this talk for fun. All right. So what is Homer and how does it work? So basically, what Homer is, it's very simple. It's a similar gene drive design, but instead of just targeting a gene with a guide RNA and encoding Cas9, we're also targeting an essential gene now, and we're encoding a rescue in the gene drive. So now the gene drive has a ability to rescue the lethality that's caused by a guide RNA and Cas9 targeting an essential gene. So we picked this gene called polymerase gamma 35, which is required for replication and repair of mitochondrial DNA. And we've designed 2 guide RNAs to target this initially to see how well this works. And so it's just a guide RNA that's encoded in the fly. And then we cross it to a Cas9 expressing line just to see how effective the guide RNA is. Well, it turns out that we actually designed 2 guide RNAs, and one of our guide RNAs worked exceptionally well. It killed everybody. So when you encode the guide RNA and the Cas9 in an animal, the animal is dead because you're targeting an essential gene, and that kills the organism. Our other guide RNA didn't work that well. It actually, if you see these circles, it actually, it worked moderate, it just didn't, we didn't see 100% killing. We saw a lot of survival happening from that guide RNA. So this told us that, again, not all guide RNAs are created equal, because these guide RNAs are inserted in the exact same location in the genome and the fly, and they use the same promoters, and we're crossing to the same Cas9 line, but one guide RNA works a lot better than the other. And I just want to point this out, the mechanism of how we get 100% killing here is that when we cross our guide RNA to Cas9, the Cas9 is coming from a mother. So the mother is maternally depositing Cas9 into all of her embryos. And then the Cas9 is also expressed zygotically throughout development. And the guide RNA is also expressed zygotically. And so what ends up happening is as the organism is developing from an embryo into an organism, during development, you get continuous cleavage of the target. And what happens is some of those cells where you get cleavage, you get repair back into wild type. So it gets repaired normally. Some cells, someone's asking to control my screen. Should I reprove that?
Oh,
I'm gonna decline
yeah,
that.
you can just ignore that.
Okay.
Yeah.
Okay. Oh, asking again. Okay.
Oh, okay. If you have,
Mohammed
oh,
Zikan Abdullah is requesting remote
okay.
control of your screen.
Yeah. I'll message the
Oh,
individual.
my bad about that.
Oh,
Oh,
okay.
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 Sorry.
No worries. Okay. All right. So basically what's happening here. So when you have this continuous expression throughout development, you end up creating a mosaic animal. So some of the cells have biallelic knockouts of the gene, Cas9 cuts, and then you get repair being NHEJ, and then you get this sort of mosaic animal at the end. All right, so we use that mechanism in this system because we're encoding a guide RNA that targets a central gene, and when crossed to Cas9, you get that mosaicism, right? So the animal's basically dead unless it gets rescued, right? And so this allows us to prevent the formation of resistant alleles, because when you're targeting that gene in a location that is sort of conserved, then the only way for the animal to survive is if it gets the rescue. So the rescues, so this is the gene drive design here. We have our, basically our, you can see this polymerase gamma gene and basically the genomic architecture of it. And then you have your homology arms, your LHA, RHA, those are your left and right homology arms. You have your blue recoding in the sequence. So the guide RNA can't cut itself, but it can cut the wild type copy. And then you have your gene drive, which encodes your guide RNA and your marker, so you can track it. So basically, we built this Homer gene drive in polymerase gamma, and we have a female here that's a trans heterozygote across the different Cas9s, and then to test the transmission rate, we cross to a wild type male, and then we can measure it. So what we found is when we cross this gene drive to different Cas9s, nano, spasa, ubiquitin, or actin, we got nearly 100% transmission. So everybody that survived from that cross actually encoded the gene drive. So we forced its inheritance to 100%. And we were able to figure out that the homing rate is about 90% and the killing rate is about 10%. So that's how we achieve this 100% transmission rate. So our next question was to see, will this system work for population control? So we did this experiment in Cas9 background. We released our gene drive at 50% allele frequency, and it immediately went to 100%. And then in basically one generation, and then we watched to see if resistant alleles would take over. And we did see a few resistant alleles come forward. So these orange dots here were some resistant alleles, but they were outcompeted with the gene drive over time. So over 10 generations, we hit 100% drive individuals by generation 5, and that we watched it for 5 more generations to see if any resistant alleles would come out, and we didn't see any. And then we did another experiment here where we released our gene drive either to determine the dependence of Cas9. We released it in a Cas9 background or in a wild type background. And in a wild type background without Cas9, the gene drive does not spread. It stays around, hovers around 50, 60%. And then in the Cas9 background, it spreads to fixation. So of course, the next step is to build this system in a disease vector to enable sort of population control that's evolutionarily stable. So take home of this part of the talk, time I have. We built a Homer drive that transmits to about 99.9% in females. It's selecting a really effective guide is essential for engineering a good Homer drive. And I showed you that not all guides are created equal. We found that drive-resistant alleles can be generated, but they do not impede the spread of the drive. And that's probably because they have a fitness cost, because you're targeting an essential gene in a conserved location. So even when you get a resistant allele formed, it has a fitness cost. So we think this very simple design should be adaptable to other species. And so that's something we're working on. Again, we tested this only in flies so far. All right, so I'm going to move on to the last part of the talk. So I've talked to you so far about gene drive and engineering effector genes like anti-ZIKA and anti-dengue. I talked a little bit about the issues of gene drive in terms of, having a gene drive that's something that's controllable, confinable, safe, yet effective. A split drive may be able to provide that, but again, that may also still require a significant amount of oversight, regulation, governance, public acceptance. All these things need to happen. It's still a gene drive. It still can spread into a population, which has its own set of issues. So with that, we've been thinking about how can we use CRISPR in another way to control a population that gives us complete control, right? And not something that's expected to persist in the environment or spread. And so we came up with this here. It's based off this very old accepted technology called SIT, which is sterile insect technique. It's a method of biological control of animals, not only mosquitoes, but it's used a lot for insects. And the way this works is really simple. I mean, this dates back to like the 1950s. What you do is you macerate your insect of choice in a factory. Then you use irradiation to irradiate. Well, first you separate the males and then you irradiate them. And then you deploy these irradiated males into a population, and they're going to go find their females, mate with them. And those females are not going to produce progeny because they just mated with a sterile male. And in insects, a lot of the males actually, for a lot of insects, the males actually deposit things with the sperm into the female that sort of prevents the female from wanting to mate again, right? So these females don't give progeny after mating with these sterile males. So the way this works is you just inundate a population with as many sterile males as possible, and you can suppress. And on the bottom here are a few examples of this technology that's been successful, like the screwworm, for example, the Mexican fruit fly, the medfly. These are all things used in the United States, and this technology has been very successful at controlling them. It does have some issues, and one of the issues is that the irradiation can reduce the fitness of the released males. When you're irradiating, you're kind of randomly messing up the DNA, like you're causing mutations, or deletions, and inversions, things like that affect the fitness of the animal. It doesn't work. This system doesn't work well for mosquitoes because the mosquitoes are very fragile, and the radiation really affects their fitness. And final thing is that you have to release adults for this, and which makes it very difficult to scale. And this technology is a numbers game. The more sterile males you get out there, the more effective the technology is. So there's this, the best example of this technology is a classical example is the eradication of the screwworm. So the screwworm is this insect that has this larvae that basically it lays its larvae in cattle or livestock, and the larvae actually burrows into the skin and consumes the flesh. So it's a flesh-eating larvae, and then it comes out as a fly. And so this has been really problematic for like the livestock industry. I mean, these could also do the same thing to humans, but of course it would have to burrow into your skin and stuff like that. But you can't find examples of this affecting humans. So in the 1950s, this of course is a big problem in the United States. But what they did is they released these sterile males and throughout the United States, and they were able to eradicate it, the species using a sterile insect technique. And then they continued on through all of Central America, all the way down to Panama, where today there's a permanent barrier in Panama where they continually release 15 million sterilized screwworms per week to prevent the screwworms from coming back up through South America back into Central America and then back to the United States. So this is a permanent barrier they've created. So you can see like how expansive in range this approach has been useful for and how effective it's been. And there's a good article in The Atlantic by Sarah Zhang, which talks about this, which came out in 2020. It talks about how this is done. You can take a look at that. So what we were thinking of is sort of how can we sort of revisit this concept for mosquitoes. And the existing technologies using SIT for mosquitoes, there's a few of them. Not using radiation, but using other ways of sterilizing mosquitoes, like Wolbachia or genetic sterilization. They rely on releasing adults. So releasing adults is problematic because it requires you to have a factory very close to the release site, and so it's very costly to build all these factories all over the world, and you have to manually go out and release the adult males. So what we were thinking of is, can we generate a facility to enable us to deploy eggs? And the nice thing about deploying eggs for some mosquito species is that the eggs actually diapods. So you can actually store the eggs for up to a year as an egg and then drop it in water and out comes your mosquito. It's amazing. It's kind of like a seed. And so if you could have a factory that generated eggs, then you could sort of hopefully drone them out into field sites and sort of reduce costs and increase scalability in that way. All right, so we of course wanted to use CRISPR to do this, and so we came up with this idea, which we call precision guided SIT. It's been published. Here's 2 publications that came out in Nature Communications, one very recently in Mosquitoes. And basically the way this works is very, very simple. You basically engineer 2 different strains of mosquitoes. The first strain is a guide RNA line. And what that is a mosquito that encodes multiple guide RNAs that target essential genes. And those are genes important for female viability and genes important for male fertility. And then you engineer another strain of mosquitoes that encode Cas9 that express both in the germline and the soma. So separately, these 2 strains do nothing, right? Because Cas9 needs a guide RNA to know where to cut. And the guide RNA needs Cas9 to do the cutting. So if you maintain these lines separately, there's nothing happening in the organism. But when you cross them together, then the magic happens. So if you homozygose both these strains, so that all progeny that come off are going to be transheterozygous, then essentially you can ensure that you only will result in a sterile male coming off. Because the Cas9 and guide RNA will come together and target these genes important for female viability or male fertility and create a mosaic animal that is essentially a sterile male. And this gives us a lot of features that are interesting in terms of logistical scalability, the ability to release eggs. It also gives us the feature of being environmentally friendly. We don't have to rely on insecticides. We can target a single species and suppress it. It's also evolutionarily stable because, again, this is all done in a facility. So you have a facility, you maintain the 2 strains, you take the females from one, the males from another, you robotically sex sort them, put them into another cage for them to mate and then produce progeny. And then you release them, and the end product is a dead end. It's a sterile male that doesn't reproduce in the environment. This makes it very safe. It also makes it effective because it uses this old concept of sterile insect technique, and it's controllable, right? You can control the system. A lot different than gene drive. So what have we done so far with this? So we've engineered it in a few different species so far: Drosophila melanogaster, Drosophila suzuki, Aedes aegypti, Anopheles gambiae. In Aedes aegypti, we're able to sterilize 100% of the males that are derived. And also we have a system that can convert all the females into intersex males. And we have a separate system that makes all females flightless, basically. So on the bottom here, this is our system that converts them to intersex. Intersex is basically a female that's converted into a male because we're targeting genes that are important for sex determination. So you have a wild type male wild type female, and then you have this intersex female. So it has a very long maxillary pulp and it has male-like genitalia. And these intersex individuals are sterile, which is very useful. So let's see. I have a video that I wanted to show you as part of my last slide. So basically, it plays a song. So what you're looking at here, these are actually, can you guys hear me talking? Or is this too loud? Can you hear me? Okay, so basically what you're looking at here is, these are Aedes aegypti mosquitoes that have just hatched. These are females, and they're sitting on the water because they're flightless. They can't fly off the water. And when you do this cross and you hatch the line, all the females look like this. And so here's them. We move them onto a little tray. You can see them, they can move their wings around, but they can't actually, they're not strong enough to fly. And part of the reason why they're not strong enough to fly is because we're targeting a gene, a muscle gene that's important for flight, but only in females. The males can actually fly perfectly fine. Just knocking out this gene doesn't affect their flight behavior at all. And so this is very powerful if you can make females unable to fly like this, and they're not going to be able to bite people. They're not going to be able to mate because they mate in swarms. They're not going to be able to transmit pathogens. And so we think that this is going to be very powerful for genetically controlling them in the field, either by knocking out their ability to fly or converting them into males or just killing them altogether, which is another thing we're working on. It turns out killing just the females is harder than one might think. But doing this is actually a little easier. So we're working on ways of just killing them direct altogether. So how would one implement this pgSIT approach? So what we're thinking is that you'd have a production facility that would sort of mass rear the two strains. You would use some sort of automated sex sorting device. Companies have generated these already. Verily has a really nice system, and this is a picture of Verily's system here that they've shared. There's also Senecio Robotics that have generated very nice robotic sex sorting systems. So you can sex sort them, you can cross them, you can accumulate the eggs. Again, these eggs can survive up to a year. But then you can simply just deploy them into the environment. And when you drop them into water, only a sterile male will come out. The females will either be flightless, or they'll be converted into males, or they'll be dead. And if we use the flightless technology, we can use a box like this where the males have to fly out. So the flightless females will never make it into the environment. And I wanted to point out at the bottom here is that this approach is going to require multiple releases. So you have to release, basically every week, basically, until you suppress and eradicate a population. And on islands, like, shown here is French Polynesia, that islands present a perfect opportunity here for genetic control because they're confined and population sizes are small. So if you got a technology like this and deployed it, we believe you can actually eradicate the disease vector or the burden from that location. And this could be useful for Aedes aegypti, but also for Anopheles gambiae, which is a major vector of malaria in Africa, or Anopheles stevensii, which is a major vector of malaria in India. So you can adapt this to other species, the platform technology. We're really excited about this. We work on gene drive too, but gene drive is really interesting once you start working on it in the lab, and you can see that you can get 100% transmission. It's interesting, but I think it has these other issues of taking away our control. And I think that's something that is a little bit scary because you can think of scenarios where losing control could be a problem. And so you don't want to lose control. So going forward, our lab, we're working on other things besides mosquitoes too, but with the mosquito side of things, we're going to continue working on developing anti-pathogen effectors. We're really excited about using CRISPR RNA targeting systems in mosquitoes that could potentially build something like a Trojan horse, where you can actually have the system expressed, but only activated in response to a virus and potentially kill the organism. Of course, we're going to continue working on confinable gene drive systems like the Homer system and linking our effectors to that drive, and then exploring opportunities for trying this in the field. But again, we'll have to see how things evolve there. And then moving forward as fast as we can with precision-guided SIT, we think this technology is going to be very impactful. It could probably be used relatively quickly by gaining public acceptance and regulatory approval. And if we can do that, then we think we can save lives and prevent transmission of many pathogens by mosquitoes. Oops, I skipped my last slide. This is my last slide here. Of course, I want to thank everybody in our lab, all our students, techs, undergrads, the people doing the work, all of our collaborators and funders on all these various projects. And yeah, I guess I can open it up for questions.
Thank you so much, Dr. Akbari. That was an incredible talk, full of real-world applications, a lot of very cool research going on. And yeah, we're definitely opening the floor up to questions now. I see that we already have a question in the chat from Fatima. Great talk, thank you. For Homer, given the gene drive efficiency is at 60 to 70% in non-Cas9 backgrounds, what are your thoughts on Homer and other split drives' ability to affect the desired outcomes in natural populations? Dr. Akbari, I don't know if you wanted to comment on that.
Oh, that's a great question, Patrima. Yeah, I think, given that Homer is able to basically, by targeting an essential gene, you can ensure the transmission of Homer. And we got, almost 100% transmission in many of our different Cas9 backgrounds. We think the Homer system is going to be very useful for population control in the wild. And of course, we designed our system to be a split drive. And we did that because we want to try to have some control of the system. We don't want to design a system that's going to just, spread super easily into a population, because we think that, I just don't think the world is ready for something like that at this point, especially given the current pandemic we're in and all these issues. I think we want to have control. And so I think going forward, if you compare like the Homer system to just a regular split drive system that doesn't target an essential gene, the Homer system is far more effective because you're forcing the transmission by killing everybody first and then rescuing the ones that receive the Homer allele. And you're also doing that by using the homology directed repair in the germline to copy over. So I think that approach is definitely the best approach to date for population modification. And so I think, if we're going to see a gene drive in the field, it's going to be something like that. If we're going to rely on population modification, it's unclear, what is wanted. So like I showed you in the talk, there's two approaches, right? Population suppression, population modification. There's a big team of run called Target Malaria that are focused on population suppression of Anopheles gambiae in Africa. And I think that's probably the more desirable outcome. Let's just suppress them. Let's get rid of them. We don't, even if they can't transmit malaria, we don't want to be bit by a mosquito. Nobody likes to be bit by a mosquito, period. So let's just get rid of them. I think the other side of the argument to that is that, well, if you get rid of it, is something else going to come and take its spot, right? Something potentially worse. So is there an ecological niche there that needs to be filled? And so that's an open question, right? And if there is one, then maybe modifying the population and having, the organism still exist, but not able to transmit might be a better approach. So these are just kind of some things that are being discussed. We don't know the answers. We need to see what happens over time in terms of, again, regulation and public acceptance and stuff like that going forward. And but in the meantime, I think it's good to think about how one could develop safer technologies that can make an impact now. I hope that answers the question.
Yeah, I feel like that was a very thorough answer to that question, Dr. Akbari. We are still open for any other questions that anyone might have for Dr. Akbari's talk. If not, I guess I myself just had a quick question about, I think, and you've mentioned this at several points throughout your talk, including just now, that public acceptance or public perception of the gene drive technology is often a significant hurdle. And I was wondering if you've developed any strategies throughout your time of working with this technology to communicate this information to the public or to persuade them of the benefits of such technology.
Yeah, I think we have actually, through presentations like this, and also sort of, we work with social scientists as well that actually help, and we hold workshops and things like that to help with engagement. But it definitely is important. You're not going to be able to please everybody, and something like with a gene drive, you may have to, it may be like the government may just have to decide how to go forward with that and make a final decision. But I think in terms of trying to persuade people, I think the best way is to educate them, explain, the problem, the current solutions, and how this can potentially make a big difference. And I think that's probably the best thing you can do. But ultimately, they're the ones that are going to decide at the end.
Yeah, that makes a lot of sense. And I'm glad to hear about all of these efforts. Great. I think we may have time for one more question if somebody has a question they would like to ask. If not, I'm also happy to hand it over to our next speaker. Well, if not, thank you, Dr. Akbari.
All right, yeah.
That was a
Thank you so
wonderful
much.
talk. And we hope
I'm going to stop my share now.
Gogec. Thank you.